In this lecture, we'll try to understand the complexity of a plant cell wall. We know that the plant cell wall is a nanocomposite crystalline cellulosic framework of a cellulose molecule which are embedded in the hydrated gel-like matrix. So we'll try to look into this simple visualization. We can see that there are fibrillar network arranged in the crisscross manner of the cellulose molecule, and we got an amorphous ground or ground matrix. So the ground matrix is a hydrated uh, gel-like, gel-like, and it contains a variety of chemicals found inside of it, which is in the very close interaction with the cellulosic fibrillar network or fibrillar meshwork of the cell wall. It contains hemicellulose, pectin, minerals, lipids, and some protein molecule. But in the secondary cell wall, it is also accompanied with the lignin, a massive amount of lignin in it. So this is going to constitute the cell wall structure. If we see, this is exactly analogous to a concrete, which having iron rods and the cementing substance, cementing material in between in the ground. If we will see the cellulosic fibrillar meshwork or framework is going to give us a tensile strength to the cell wall, while the ground matrix provide the flexibility, porosity, and help in the signaling of messages along the cell wall. This cellulosic framework having the micro and macrofibrils which are deposited in a thin concentric rings or layers, and layers after layer they are secreted towards the outside from the plasma membrane by the complex synthetic machinery. These layers are called lamellae. In a primary cell wall, these macro and microfibril lamellae are arranged in a cross helicoidal pattern, forming forming a 30 to 90-degree angle. As the arrangement or organization can be seen in plywood. In a secondary cell wall, the lamellae are more or less parallel, and they constitute a very packed layer like S1, S2, and S3 layers in the secondary cell wall. If you observe, the complex structure of the plant cell wall, here the microfibrillar framework is having a cellulosic micro and macrofibrils, which are arranged to give us very complex bundles and complex bundles which are arranged in a very sophisticated manners and giving us the three-dimensional structure of a cell wall. This arrangement organization is hierarchical, and if you see, it begins from the glucose, which is a fundamental building unit of the cellulose molecule and the bundle of a cellulose. So the glucose is in the whole cellulose molecule itself is the beta 1,4 linked glucan chains. One glucose molecule is going to link with another glucose molecule through beta 1,4 glycosidic bond to give us cellobiose, and it further grows into a long chain, and that long chain of glucosyl unit or glucose unit linked by beta 1,4 glycosidic linkage is giving us a glucan chain. The glucan chain is a unbranched chain. We'll try to see that. But the glucan chain further come together to give us a cellulose molecule as a result of intra and intermolecular hydrogen bonding, and van der Waals forces make the glucan chain together give us a cellulose molecule. The cellulose molecule are normally arranged in the form of micelles. It's called protofibrils. These micelles was the concept of a previous scientific literature, but we have just replaced it so cellulose molecule are arranged in the form of bundles, which we refer to as microfibrils. So the microfibril is the basic unit of the which are synthesized as a one unit by the cellulase synthase complex rosety on the membrane, located on the membrane of the cell. Now, the microfibrils are arranged in the form macrofibrils. The macrofibrils are the big bundles of several microfibrils when they assemble. The macrofibrils are then come and deposited in the in layer by layer manner in the structure, in the macroscopic structure of a cell wall. We are going to refer to that structure as a lamellate structure, where the layers are where one layer is placed on upon another layer in a specific way. Sometimes this layering or forming very specialized pattern to give us a three-dimensional arrangement of the cell wall. Now, beginning our story from uh, the glucose residue, we know that the glucose is the fundamental unit or the building residue of the cellulose molecule. So here, one glucose unit is linked to another glucose unit by beta-1,4 glycosidic linkage, giving us a structural polysaccharide in the form of a long unbranched glucan chain. So the glucan chain is the fundamental polymer. Normally, when we hydrolyze glucan polymers, so we can hydrolyze them partially into cellobiols, which is a disaccharide unit, and upon a complete hydrolysis of the glucan chain, hydrolyze into a glucose molecule. Uh, Now, the glucan chain having intermolecular hydrogen bonding and a van der Waals forces. Especially, this is hydrogen bonds are formed between the carbon hydroxyl of carbon number 3 and oxygen of the next glucose unit to give us intermolecular hydrogen bonding and a structure of a cellulose molecule. In the cellulose molecule, we have a large number of glucose monomers. Normally, the cellulose contains 500 to 15,000 of these monomeric units, and usually they have a length of 0.25 to 8 micrometer long. Sometime the cellulose complex is the combination of several glucan polymers. Now we know from 40 to 70 of the glucan polymers combined together to give us a cellulose molecule. But this actually, the structure of the cellulose is maintained not only by the intermolecular hydrogen bonding of the glucan chain in itself, but there are several intermolecular hydrogen bonding between the adjacent glucan chain, and the glucan combined together. The glucan chain combined together to give us a complex structure of a the cellulose molecule. The glucose chain, the glucan chain are showing polarity in the cellulose molecule. So all the reducing ends are actually facing towards the one side in the structural organization. Now, the cellulose molecule combine together in a different number to give us a fundamental tiny crystalline in a non-crystalline domain cellulose fibrils, which is the fundamental unit of the structural unit. From 6 to 10 glucan chains are combined together, and they are giving us a fundamental unit of the cellulose compound, which is called micelles or elementary crystalline unit. They have a diameter of 2 to 3 nanometer wide lattice form. As a result of crystallization process in a tiny crystalline domain, in the modern literature, we are considering the microfibril as a basic or fundamental unit. So normally the microfibril is formed by the micelles. Three micelles combine, or elementary crystalline units combine to give us the microfibrils. The microfibrils are the fundamental structural unit of this cellulosic framework in the plant cell wall. The microfibril can be seen or it can be visible with electron microscope. And these fibrils, a bundle of lattice where one glucan chain is having a hydrogen bonding with adjacent glucan chain. Now, this is a structural unit of the cellulose molecule. It's synthesized as a one unit by the cellulase synthase rosity complex found on the plasma membrane of a cell. Normally, it is having a diameter of 3 to 5 nanometer, and the length is more than 10 micrometer. In each microfibrillar unit, there are 3 micelles. So the microfibrils can be considered as a supramolecular cables or rods or ribbons formed as a result of as a result of combination of cellulose molecule. Now, per microfibril, there are about 18 to 36 cellulose molecule. In a cross-section view, if we examine, so the total number of 18 to 36 cellulose molecule can be found in the one microfibrils. These microfibrils are synthesized by the complex machinery found on the complex machinery of cellulose synthase protein, which is the part of the cellulose synthase rosity complex found on the plasma membrane of a cell. The microfibrils are combining together or packed together to form a big size, microscopic size bundles, which are known as the macrofibrils or fibrils. And they're basically appear to be a cellulosic. Fibrils are bundles of microfibrils, which contain several hundred of the cellulose, from 100 to 1,000 of the cellulose can be found in the single macrofibrils, and this is more than 100 nanometer in diameter and a big structure, which is very tightly packed in the secondary cell wall and having a splitted arrangement or organization in the primary cell wall due to cell wall expansion. Due to the macrofibrils are arranged in the lamellate fashion, and lamellae are placed one upon another in a very specific manner or organization uh, to give us the macroscopic structure of a cell wall. Now, if you're trying to look into the whole synthetic process, that how these microfibrils are synthesized biologically, and these microfibrils are synthesized as one unit by a specialized machinery located on the plasma membrane of a cell. And this machinery is not actually static machinery. It's a dynamic machinery moving all the way in the membrane. And if we see, and the cellulose synthase complex is arranged in the form of a rosity. It's a membrane bounded at a catalytic machinery, and each of this is about 25 nanometer in diameter, which is a big complex. And we know that the component of the machinery are actually synthesized by the Golgi bodies, and they are actually coming into the membrane and becoming a part of the membrane. Now, cellulose synthase complex rosity is having a hexameric structure with 6 lobes in it. Each lobe by itself is a trimer, of the CESA, which is a specialized transmembrane glycosyl transferase protein, which is known as the cis A protein, or which is known as cellulase synthase A isoform protein. Each of their trimer contain three of the CSA or cellulose synthase protein, and we have 18 cis A in the cellulose synthase complex machinery. Now, how the cellulose are synthesized? Now, the precursor of the cellulose molecule in the energized or active form is coming from a biosynthetic machinery of cytosol in the form of UDP glucose molecule. The UDP glucose, and this glucose molecule is coming from the cytoplasm and entering into this cellulase synthase complex machinery from the cytoplasmic phase of the this machinery, and as they enter into this catalytic machinery, they are combined together. The UDP glucose are combined together, and from the UDP glucose, they are joined. The glucose are joined through beta 1,4 glycosidic bond, and as a result, what we're getting beta 1,4 linked glucan chain are coming out to the toward the exterior and deposited. So this beta 1,4 glycosidic bond, or the beta 1,4 linked glucan chain, are extruded toward the outside of the plasma membrane, and they, through a self or spontaneous crystallization process, combine together to give us through a self assembly mechanism, all the beta 1,4 linked glucan chain are combining together to give us a fundamental unit of the cellulosic fibrillar network, or machinery, or network, or a framework of the cell wall, and that is known as microfibril. As we know that there are 18 cis A protein. That's why we have an 18 glucan chain as universally accepted number of. So as the microfibrils are coming out, are deposited towards the exterior, so this is actually becoming a part of ground. But the ground is filled by the pectin and hemicellulose secreted through a vesicle of a Golgi bodies, which forming the scaffold of this microfibrillar cellulose chain. The cellulose molecule is a semi-crystalline structure, which means that it contains, and it is not 100%. There is a non-crystalline or amorphous region alternate with the crystalline region and the cellulose microfibrils. According to another point of view, the cellulose is having its crystalline core surrounded by a non-crystalline or amorphous glucan chain, where the glucan chain in the crystalline region are arranged in the well packed in a compact manner. Non-crystalline region, the which are surrounding the core of the crystalline region, are actually loosely arranged glucan chain, and they are very reactive and they're very reactive and exposed to the action of several enzymes and water activities on it.